The Gene, An Intimate History by Siddhartha Mukherjee. Picture a family secret for just a moment. Not the gossip that bubbles up at holiday dinners, but something deeper, something woven into the very fabric of your bloodline. A shadow that reaches back through generations, touching uncles, cousins, and maybe even you. This is where our journey begins. Not in some gleaming laboratory, but in Calcutta, India, with a young doctor named Siddhartha Mukherjee visiting his cousin Moni. Mani lives confined to a mental institution, lost somewhere in the fog of schizophrenia, and as the author looks at his cousin, he sees something more than just a personal tragedy. He sees a pattern emerging from the mist. He remembers his uncle Rajesh, the most brilliant of 5 brothers, whose dazzling mind slowly unraveled into manic depression, his life cut short at just 22. He remembers another uncle, Jagu, who wandered through life haunted by visions and phantom voices, eventually diagnosed with schizophrenia himself. Madness, it seems, has been flowing through the Mukherjee family bloodline for generations, and this isn't just one family story, it's a deeply human one. It's the story of heredity. It raises the oldest question we've ever dared to ask. Where do we come from? Why are we the way we are? Are we the authors of our own lives? Or are we simply actors reading from a script, written in a language we can't even understand? Today we're diving into a book that wrestles with these profound questions. It's called The Gene, An Intimate History, written by Pulitzer Prize-winning author and physician Siddhartha Mukherjee. This isn't just another science book. It's a sweeping epic, a detective story, and an intimate family memoir all woven together. It has captivated millions of readers worldwide because it does something remarkable. It takes the most complex and important scientific idea of our time, the gene, and transforms it into a breathtaking story about what it means to be human. Over the next hour, I'm going to guide you through this incredible history. We'll journey from an obscure monastery garden in the 1800s to the darkest corners of Nazi Germany and into the thrilling race to crack the very code of life itself. My promise to you is this: by the end of our time together, you won't just understand what a gene is. You'll understand the extraordinary power it holds over our past, our present, and most importantly, our future. You'll discover that this isn't a story about them. It's a story about us. For most of human history, the idea of heredity was shrouded in pure mystery, a realm dominated by superstition and wild guessing. The ancient Greeks, for example, had some fascinating theories. Pythagoras, the famous mathematician, believed that a father's semen traveled through his entire body, collecting invisible vapors from every part, capturing color from his eyes, texture from his skin, and delivered this complete, miniature blueprint to the mother. The mother, in his view, was merely the soil, the warm incubator, while the father provided the all-important seed. Aeschylus, the playwright, even weaponized this idea in a courtroom drama, arguing that killing one's mother wasn't really so terrible because she was, technically speaking, a stranger, just a nurse for the seed. It was a convenient theory, especially if you happened to be a man. Then came Aristotle, a thinker who valued evidence over convenient narratives. He spotted the obvious flaw. Children clearly inherit traits from their mothers too, and traits can mysteriously skip generations. A boy might be born with his grandmother's distinctive nose, even if neither of his parents carried it. Aristotle became the first to grasp that heredity wasn't about passing down physical material, but about transmitting information. He called it a message, or a principle of movement. The father and mother both contribute instructions, which then guide the formation of a child. It was a revolutionary insight, centuries ahead of its time. He had essentially imagined the gene without ever knowing it existed. For 2,000 years, these ideas spun in circles, going nowhere. Some believed in a homunculus, a tiny, fully formed human curled up inside every sperm, just waiting to inflate like a balloon in the womb. Others thought traits simply blended together, like mixing red and white paint to create pink. But no one could explain the precise rules governing inheritance. Heredity remained a science without laws. And then, in the 1850s, in a quiet monastery garden in what is now the Czech Republic, a humble monk began growing peas. His name was Gregor Mendel. Mendel wasn't a world-famous scientist. He was a modest, curious man who had actually failed his teaching exams in biology not once, but twice. Yet he was patient, meticulous, and a natural gardener. He wasn't trying to unlock the grand secrets of the universe. He simply wanted to understand why, when he crossed a tall pea plant with a short one, he didn't get a medium-sized plant in return. For 8 painstaking years, Mendel carefully cross-pollinated thousands of pea plants. He tracked 7 distinct traits: plant height, seed shape, flower color, and others. And through this tedious, repetitive work, sow, pollinate, pluck, shell, count, repeat, he uncovered the fundamental laws of heredity. What he discovered was revolutionary. Traits didn't blend at all. When he crossed a tall plant with a short one, all the offspring were tall. The tallness trait was dominant, and shortness was recessive. The short trait hadn't disappeared. It was just hiding, lying dormant. When he then bred those tall offspring with each other, the shortness trait reappeared, perfectly intact, in about 1/4 of the new generation of plants. This could only mean one thing. Heredity was passed down in discrete, indivisible units. Each parent contributes 1 unit for each trait to its offspring. These units, these particles of information, remained separate and whole, never blending together. They could be dominant or recessive, but they were always there, always intact. Mendel didn't have a name for them yet, but he had discovered the gene. In 1865, he presented his groundbreaking findings to a small group of local farmers and scientists. They were completely mystified. All his numbers and ratios seemed like arcane numerology to them. His paper was published in an obscure journal, and then, for nearly 40 years, it was completely forgotten. The key to one of life's greatest mysteries lay buried in plain sight, gathering dust on library shelves. While Mendel's paper was being ignored, another giant was wrestling with the same problem from a completely different angle. In England, Charles Darwin had just published On the Origin of Species. He gave the world the magnificent theory of evolution by natural selection. He explained that life evolves through 2 elegant principles: variation and selection. Animals within a species are not identical. They have natural variations. And in the constant struggle for survival, those with favorable variations, the fittest, are more likely to survive, reproduce, and pass on those advantageous traits. It was a brilliant explanation for the grand sweep of life on Earth. But Darwin knew there was a gaping hole in his theory. He couldn't explain how traits were actually passed on, or how new variations arose in the first place. He believed in a blending inheritance, where traits from parents mixed like paint. A sharp-minded engineer named Fleming Jenkin pointed out the fatal flaw. If traits truly blended, any new, favorable variation would be diluted into extinction within just a few generations. It would be like dropping a single spoonful of black paint into a giant vat of white paint. The result isn't a bold new color. It's just a slightly gray or white. Darwin's theory desperately needed Mendel's particles, but he never knew they existed. In a heartbreaking twist of history, Darwin actually owned a copy of a book that mentioned Mendel's work, but he scribbled notes on every page except the one that discussed the pea experiments. The two most important biological ideas of the 19th century were like ships passing in the night, never meeting. It wasn't until the year 1900 that three different scientists working independently across Europe stumbled upon Mendel's forgotten paper and recognized its genius. Suddenly, the science of heredity exploded into vibrant life. An English biologist named William Bateson became its fiercest champion. He gave the new field a name, genetics. And in 1909, a Danish botanist named Wilhelm Johansen finally gave Mendel's unit of information its own name, the gene. The gene was an idea of incredible power, but like all powerful ideas, it cast a dark shadow. Darwin's own cousin, a brilliant but troubled man named Francis Galton, became obsessed with the idea of improving the human race. He reasoned that if farmers could breed better cows and pigeons through artificial selection, why couldn't humanity do the same for itself? He coined a term for this new science, eugenics, from the Greek words meaning good in birth. Galton believed that not just physical traits, but also intelligence, talent, and even moral character were inherited. He wanted to encourage the best people to have more children and discourage, or even prevent the unfit from reproducing. It started as a utopian dream, a plan to give the more suitable races or strains of blood a better chance of prevailing. But this dream rapidly morphed into a nightmare. In the early 20th century, the eugenics movement swept across America and Europe like wildfire. It wasn't a fringe idea. It was mainstream science, supported by leading thinkers, politicians, and social reformers. In America, Better Babies contests were held at state fairs, where infants were judged and scored like livestock. The movement was fueled by anxieties about immigration, social class, and fears of race degeneration. The twisted logic of eugenics reached its most terrifying conclusion with the story of a young woman named Carrie Buck. In 1924, Carrie, a poor teenager from Virginia, was committed to the state colony for epileptics and feeble-minded after giving birth out of wedlock. Her mother, Emma, was already living there as a resident. The state of Virginia, led by eugenics-obsessed officials, wanted to sterilize Carrie to prevent her from passing on her alleged feeble-mindedness. Her case, Buck versus Bell, went all the way to the Supreme Court. The evidence against her was flimsy and fabricated. A social worker described Carrie's infant daughter, Vivian, as having a look about it that is not quite normal. That vague impression was deemed sufficient evidence. In 1927, in an infamous 8-1 decision, Justice Oliver Wendell Holmes, Jr. wrote the majority opinion, declaring, "It is better for all the world if instead of waiting to execute degenerate offspring for crime or to let them starve for their imbecility, society can prevent those who are manifestly unfit from continuing their kind." He concluded with one of the most chilling lines in American legal history: 3 generations of imbeciles is enough. Carrie Buck was sterilized on October 19, 1927. Her daughter, Vivian, later proved to be a perfectly normal, even bright student, before dying tragically young. Carrie's case opened the floodgates. By the 1930s, over 30 states had compulsory sterilization laws, and tens of thousands of Americans deemed unfit were robbed of their ability to have children. American eugenicists were seen as pioneers, and in Germany they had some very attentive students. the Nazis took the ideas of eugenics and twisted them into the brutal doctrine of racial hygiene. Nazism, one of its proponents declared, was nothing more than applied biology. In 1933, the Nazis passed their own sterilization law, modeled directly on American laws. But they applied it with ruthless, industrial efficiency. They sterilized hundreds of thousands of people deemed to have hereditary diseases, schizophrenia, epilepsy, blindness, deafness, and even alcoholism. The propaganda machine went into overdrive, with films and posters portraying the genetically unfit as parasitic burdens on the state. Sterilization was just the first step. The journey from labeling lives as not worth living to actually taking those lives was horrifyingly short. In 1939, the Nazi euthanasia program, codenamed Action T4, began its grim work. At first, it targeted disabled children, but soon expanded to adults. People were taken to specially designed killing centers, murdered with carbon monoxide gas, and their deaths were disguised as natural illnesses. This program became the training ground, the methodological blueprint for the Holocaust itself. The logic used to justify the murder of the mentally ill was seamlessly extended to the systematic extermination of Jews, Roma, and others deemed racially impure. The gene, an abstract idea of heredity, had been forged into a weapon of mass murder. The horrors of Nazi Germany cast a long, dark shadow over the entire science of genetics. It exposed the terrifying danger of conflating genes with identity, and identity with human worth. While the world was tearing itself apart, the scientific quest to understand the gene continued, but it shifted from the abstract to the physical. What actually is a gene? What is it made of? For decades, scientists assumed genes must be made of proteins. Proteins are incredibly complex molecules, capable of a staggering variety of functions, so it seemed perfectly logical they would carry the complex information of life. There was another molecule found in the cell's nucleus, dismissively called a stupid molecule, by one biochemist named deoxyribonucleic acid, or DNA for short. It seemed far too simple, made of just 4 chemical building blocks, or bases, adenine, guanine, cytosine, and thymine, abbreviated as A, G, C, and T. Most scientists thought it was just structural scaffolding, biological filler. The first real clue that this stupid molecule was far more important came from a quiet, shy scientist named Frederick Griffith back in the 1920s. He was working with 2 strains of pneumonia bacteria, a smooth, deadly strain and a rough, harmless one. He discovered that if he killed the deadly bacteria with heat and then mixed their cellular remains with live, harmless bacteria, something astonishing happened. The harmless bacteria mysteriously transformed into the deadly kind. Some transforming principle, some chemical substance, had passed from the dead cells to the living ones and permanently changed their hereditary nature. The identity of this transforming principle remained a tantalizing mystery until the 1940s, when a meticulous bacteriologist at Rockefeller University named Oswald Avery took up the challenge. For over a decade, Avery and his dedicated team painstakingly purified the mysterious substance from the dead bacteria. They treated it with enzymes that destroyed proteins, fats, and sugars. Nothing stopped the transformation. But when they used an enzyme that specifically destroyed DNA, the transformation stopped cold in its tracks. The conclusion was inescapable, though Avery himself was almost too cautious to say it out loud. The genetic material, the true carrier of hereditary information, was DNA. The discovery that genes were made of DNA launched a frantic, competitive race to understand its structure, If form equals function, then the precise shape of the DNA molecule had to hold the secret to how it actually worked. This race featured a cast of brilliant, difficult, complicated characters. In London, at King's College, there was Maurice Wilkins and Rosalind Franklin. They were using a sophisticated technique called X-ray crystallography to take pictures of the DNA molecule. Franklin was a brilliant, fiercely independent scientist, working in a world utterly dominated by men. She and Wilkins had a famously frosty relationship, a hate at first sight, that seriously hobbled their collaboration. But Franklin's work was extraordinarily meticulous. She produced the clearest X-ray images of DNA ever captured, including a famous one known simply as Photograph 51. The image, with its distinct X shape, strongly suggested a helical, or spiral, structure. Meanwhile, across the English Channel in Cambridge, two young upstarts were trying to solve the structure not through painstaking experiments, but by building physical models, like assembling a complex three-dimensional puzzle. They were James Watson, a brash, ambitious young American, and Francis Crick, a brilliant, talkative British physicist who was still technically just a graduate student. They were a perfect, if unlikely, pair. They knew the world's greatest chemist, Linus Pauling, was also racing toward the answer in California, and the pressure was intense. Watson and Crick's first attempt at a model was an embarrassing disaster. They built a triple helix with the sugar phosphate backbone on the inside. When Rosalind Franklin saw it, she methodically tore it to shreds with her sharp, devastating critique. Deeply humiliated, they were ordered by their superiors to stop working on DNA entirely. But the story took a dramatic, controversial turn. In a now infamous moment, Maurice Wilkins, without Franklin's knowledge or permission, showed Watson her Photograph 51. For Watson, it was like being struck by lightning. "The instant I saw the picture," he later wrote, "my mouth fell open and my pulse began to race." The distinctive X shape confirmed it absolutely had to be a helix. Another crucial piece of data, also shared without Franklin's full knowledge from a report she had written, gave them the precise dimensions they needed. Armed with these stolen clues, Watson and Crick returned to their model building with renewed energy. The final, crucial insight came to Watson on a Saturday morning. He was casually playing with cardboard cutouts of the four bases, A, T, G, and C. He suddenly realized that an A base on one strand could form a perfect chemical bond with a T on a second strand, and A, G could pair perfectly with a C. They were complementary partners, like puzzle pieces. This was the secret, the key to everything. DNA was not a single helix or a triple helix, it was a double helix. Two strands spiraling gracefully around each other, like an elegant, twisted ladder. The sugar phosphate backbones formed the outer rails of the ladder, and the paired bases, A bonding with T and G bonding with C, formed the rungs connecting them. The structure was so beautiful, so elegantly simple, it had to be right. It immediately explained everything. The sequence of bases along one strand, AGGTCA and so on, could carry a code, a molecular language of life. And because the two strands were complementary mirrors of each other, the molecule could unzip down the middle, and each strand could serve as a perfect template to make a new copy of its partner. It explained brilliantly how genetic information could be both stored and precisely replicated. In April 1953, Watson and Crick published their historic one-page paper in the journal Nature, modestly concluding with the understated line, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." It was one of the greatest understatements in the entire history of science. The discovery of the double helix marked the end of one era and the dawn of another. The question was no longer what a gene was, but how it actually worked. How did the simple four-letter alphabet of DNA translate into the rich, staggering complexity of life? The answer came together in pieces, like assembling a vast puzzle. Scientists discovered that the DNA sequence in a gene is first transcribed into a messenger molecule, a single-stranded cousin of DNA called RNA. This RNA message then travels out of the cell's nucleus to a cellular machine called a ribosome, where it is translated into a protein. This elegant flow of information from DNA to RNA to protein was dubbed the central dogma of molecular biology by Francis Crick. Genes, it turned out, are essentially recipes for making proteins. And proteins are the true workhorses of the cell. They function as enzymes that catalyze chemical reactions, structural components that give cells their distinctive shapes, and hormones that carry vital signals throughout the body. Everything from the color of your eyes to the intricate way your brain processes a thought is governed by proteins, which are in turn governed by genes. Scientists also painstakingly cracked the genetic code itself. They discovered that the ribosome reads the RNA message in three-letter words called codons. The codon, August, means start here and add the amino acid methionine. The codon, GGU, means add glycine, and so on for all 20 amino acids that serve as the building blocks of proteins. A gene, then, is a long sentence written in these three-letter words, spelling out the precise sequence needed to construct a protein. A single mistake in this genetic sentence can have absolutely devastating consequences. The disease sickle cell anemia, for example, is caused by just a single letter change in the gene for hemoglobin, the protein that carries oxygen in our blood. Just one A is swapped for a T. This tiny molecular typo changes one amino acid in the protein, causing the hemoglobin molecules to clump together abnormally, warping red blood cells into a rigid, sickle shape that can block blood vessels and cause excruciating pain. It's a powerful, tragic illustration of the central dogma in action. One tiny change in a gene leads to one change in a protein, which cascades into a life-altering disease.